Hello, welcome to Cambridge O-Level YouTube channel. If you can remember, in previous presentations, we discussed about photosynthesis and we discussed about the starch test. Today onwards, we are going to talk about the factors necessary for the photosynthesis. If you can remember, just memorize the factors necessary for the photosynthesis. That is carbon dioxide, water, chlorophyll, and sunlight. So today we are going to check whether carbon dioxide is necessary for the photosynthesis. As you know, plants use carbon dioxide as a raw material to make food. So they are absorbing these carbon dioxide from tiny pores in the leaves. To them we are calling as stomata. So this will be helping to build up glucose inside the plant. Somehow this glucose will be turning into starch. In previous lessons, we discussed about that process. As a byproduct of photosynthesis, it will be producing oxygen. The overall process is summarized as shown in this equation. 6 carbon dioxide, 6 H2O. When there is sunlight and chlorophyll, it will be producing glucose, C6H12O6 and 6 oxygen molecules. We'll move to the experiment. These are the materials needed. We need a healthy potted plant. Better to have 2 healthy potted plants, and we need 2 transparent plastic bags and a small container with potassium hydroxide or soda lime. We are using it to absorb carbon dioxide. In this experiment, we are going to limit carbon dioxide. So in that case, the plant will be lacking with carbon dioxide. If it doesn't have carbon dioxide, it means if it is lacking with carbon dioxide, can it do the photosynthesis? No. They will be reducing the rate of photosynthesis. Somehow it will be not doing the photosynthesis. Okay. Rather than that, we need rubber bands or threads. So if you can remember the discharging process, that is the major step in this photosynthetic experiment. Discharging means we have to keep the plants in dark place around 24 to 48 hours. It means 1 to 2 days. This experiment, actually this process is doing to remove all the starch already stored inside the plant. Plants may already have stored starch in their leaves. If you don't remove it, you won't know whether the starch you detect was made during the experiment or was already there. So, as you can remember, in previous presentations, we discussed about the discharging process. You can go through them. Discharging means we will be removing all the starch stored inside the plant. So, after discharging the plant, we have to take, actually we need to take 2 discharge plants. So, after that, we have to take it out from the dark room and we can water the plant, and we can cover one leaf or a branch with plastic bag which contains a small container of potassium hydroxide or soda lime. As I mentioned earlier, this potassium hydroxide and soda lime have effect of absorbing carbon dioxide. So better to have potassium hydroxide or soda lime. Then the other leaf can be covered without KOH, without potassium hydroxide or soda lime. Instead, we can add sodium hydrogen carbonate. Sodium hydrogen carbonate will be releasing carbon dioxide. So we have one plant without carbon dioxide and another plant with carbon dioxide. somehow we need to tie both bags tightly to make them airtight. After keeping soda lime for the potassium hydroxide and sodium hydrogen carbonate solutions, we have to tie up both the bags. Then we have to keep these two plants on the sunlight for a few hours. After that, we can pluck two separate leaves from two plants and we can do the starch test or the iodine test. So this is the way we are going to do. We need to take two plants. These are the two plants that we have taken. All those two plants are the starch. And in here you can see saturated sodium hydrogen carbonate will be there. We can take a petri plate or a petri dish and we can add sodium hydrogen carbonate to this. And in this diagram you have mentioned the soda lime has kept inside the plant. After that, you have to tie up both the plants air tightly. If you need Vaseline, you can apply Vaseline and somehow you have to keep these two plants air tight. That is very important. So there is no any gas exchange with the external environment. So if you go through this first diagram, you can see it is saturated with sodium hydrogen carbonate, or maybe in your exam paper they might be giving it is saturated with water. Somehow there it means there are some amount of carbon dioxide in this plant. But in this plant it doesn't have any carbon dioxide. So what will happen? Can they do photosynthesis? In first plant, obviously it can do photosynthesis. The reason is it has carbon dioxide. Already there are water, chlorophyll is there inside the leaf, and sunlight is also there. But in the second diagram, water will be there, chlorophyll will be there, and sunlight will be there, but it doesn't have carbon dioxide. Why? What is the reason? The soda lime is absorbed. Already soda lime is absorb carbon dioxide. So there are no any carbon dioxide in this apparatus. So it is limiting with carbon dioxide. So it cannot do photosynthesis. It means with the time, can it produce starch? No. In this second diagram, you cannot see any starch. You can't find any starch. But in the first diagram, actually you can see starch. You can observe starch. If you do the starch test for these two separate leaves from first plant and the second plant, you can see, can observe the clear observations. So here, if you take this leaf, suppose that this leaf, we'll take this one. This leaf is from the first plant. It means first plant means this leaf. In this plant, if you take this plant, this leaf, it is taken from the first plant. It means it has already carbon dioxide. It means it has done the photosynthesis. It means there are starch. So if you do the iodine test, it means if you add iodine after doing the starch test, you can see that blue-black color. But in the second diagram, it means the second plant, if you do the starch test, you cannot see that color change. So the leaf will be remaining brown color. It means it doesn't have starch. So this is the conclusion. The leaf without carbon dioxide could not produce starch. So as I mentioned earlier, Without carbon dioxide, it cannot do photosynthesis. And if you do the starch test, it means, can you remember the starch test? So in that case, what you have to do, you have to take the leaf and you have to boil it. After that, you have to insert it to a ethanol and you have to double boil it. At that time, you can see the ethanol solution. It is colorless, but with the time it will be turning into green color and the leaf is turning into pale yellow-like color. After that, you have to take it out and resin it with water and you can add iodine solution. So that is the way you are doing the starch test. You can go through them. At that point, if it is lacking with carbon dioxide, it means no starch. It means it is not turning into blue-black color. The leaf is remaining brown color. But if there are carbon dioxide, it means in that sodium hydrogen carbonate solution, that leaf, it will be giving positive results to the starch test. It means it will be turning into blue-black color. So this experiment will be proving carbon dioxide is necessary for the photosynthesis. So that is the end of this presentation, and in next classes we will be discussing about the other factors necessary for the photosynthesis, especially sunlight and chlorophyll. Thank you for watching.